Next, we have Sara Horsman from University of Queensland talking about antimicrobial resistance and usage in dogs with skin disease and ear disease. Thanks, Sara.
Okay, thank you. So thank you so much, VetCompass, for giving me this opportunity to present today. I'll be going over the methods for uncovering the geographical associations between your antimicrobial resistances and your usage in dogs with pruritic skin and ear diseases through data linkage. So just to start, dogs with dermatological conditions affecting the skin and ears can be caused by allergies, ectoparasites, and dermatophytes. It can cause pruritus and secondary bacterial infections such as pyoderma and otitis externa. The main objective is to control the itch, and that can be achieved using anti-pruritic therapies such as glucocorticoids, cyclosporine, and ofloxacin. The secondary bacterial infections as well require recurrent antimicrobial usage, which can increase the risk of antimicrobial resistances and therefore may limit available treatment options in the future. There's been two retrospective studies, one using the VetCompass Australia database, which identified that there are lower odds of antimicrobial usage of dogs with allergic dermatitis receiving ofloxacin compared to your other anti-pruritic therapies. So what we want to know now is, has this actually had a direct result on your antimicrobial resistances cultured from those bacterial skin and ear infections in dogs? So we can achieve this using spatial epidemiology to identify and visualize your disease patterns, such as your antimicrobial resistances and usage, your spatial clusters or your hotspot areas of high multi-drug resistant isolates, and any associated risk factors. For the objective, the aim is obviously to uncover the geographical associations between your antimicrobial resistances in bacterial isolates cultured from the skin and ear infections and your antimicrobial usage in dogs treated with anti-pruritic therapies for their pruritic skin and ear diseases in Queensland. This will be achieved using a secondary data linkage approach at postcode level using a veterinary laboratory database and the VetCompass Australia database. So the hypothesis for the study is postcodes of dogs treated with a high proportion of ofloxacin will have fewer antimicrobial usages and therefore fewer antimicrobial resistances or multi-drug resistant isolates. Today, I'll go over the two study populations, the data cleaning and calculations, spatial epidemiological analyses, and the risk factor analyses methods. For both the laboratory database and the VCA database, they'll contain canine data from Queensland, Australia in 2016 to 2018 from dogs of all ages. Risk factors will also be included, such as demographics, including age, sex, breed, and neuter status, and environmental and human socioeconomic data per postcode. The laboratory database does include samples from canine skin and ear infections, bacterial culture and antimicrobial susceptibility testing results, and clinic postcodes. VCA database is veterinary consultations of your dogs with pruritic skin and ear diseases, the number of treatments prescribed, so that's both topical and systemic for your anti-pruritics and antimicrobials, and it'll be at client postcode level. For the data cleaning, for the laboratory database, I firstly removed duplicates of the same bacterial species with the same antibiogram from the same dog that was cultured simultaneously. The next step is to remove your isolates that don't have any CSLI interpretation guidelines and remove intrinsic resistances before calculating multi-drug resistances. Then I calculated the number of non-susceptible isolates per antimicrobial, in addition to your proportion of multi-drug resistant isolates, including those resistant to your medically important antimicrobials or your critically important antimicrobials. For the VCA database, obviously any consultations not pertaining to the search terms or any impossible dates will be removed, and all consultations of dogs with pruritic skin and ear conditions will be kept. The next step is to separate out your topical and systemic antimicrobial treatment types based on their active ingredients and purpose, before calculating the number of treatment types per that were prescribed, and then the proportion of dogs treated using those various treatment types. In terms of the spatial epidemiology analysis, both of the databases will have polygon maps created at postcode level in Queensland based on the proportions data. The laboratory database will include your multi-drug resistant isolates per clinic postcode. This will be all multi-drug resistant isolates combined and then broken down into the 5 most cultured isolates. For the VetCompass Australia database, it'll be the proportion of dogs with the pruritic skin and ear diseases treated using the various prescribed treatment types at client postcode level. So for instance, the proportion of dogs treated or prescribed oxaliplatin, antimicrobials, and then combinations of treatment types such as oxaliplatin and antimicrobials combined. I'll then conduct Moran's eye and local indicators of spatial autocorrelation statistics to identify any clustering or hotspot areas of, for instance, your high multi-drug resistant isolates. This will help identify, are any of the same postcodes identified as hotspots per proportion mapped visually? But to actually link the 2 databases together, I will use Bayesian conditional order regressive modeling to identify, are there actually fewer antimicrobial resistances within the same postcodes of dogs treated using oxaliplatin compared with your other anti-pruritic therapies? And also identify the associated risk factors. So for instance, is age, breed, or urban areas considered risk factors? And then lastly, just to conclude, for the veterinary laboratory database, we'll identify hotspot areas of high antimicrobial resistances, and also which veterinary clinics may actually benefit from performing routine culture and antimicrobial susceptibility testing. The VetCompass Australia database will identify hotspots of high antimicrobial usage, anti-pruritic therapies, and then are there any high usage of different combinations of treatments? And then lastly, we do hope that there is an association between the use of oxaliplatin on antimicrobial usage at postcode level in pruritic dogs to the bacterial resistances cultured from those canine skin and ear infections from those same postcodes. And this may aid in targeted antimicrobial stewardship programs. Lastly, I would just like to thank VetCompass Australia for permission to actually conduct this study and also for their extended help with extracting the database, in addition to the veterinary laboratory database, veterinary laboratory for giving us permission to use their database, and the RTP scholarship as part of my PhD for funding this work. Thank you all.
Thanks, Sarah. Fantastic.